Western Sydney University Library extends a warm welcome to the Thought Leadership Event Series number 20. We proudly present talks by Western Sydney University researchers whose work aligns with the United Nations Sustainable Development Goals, also known as SDGs, celebrating our number one ranking for commitment to SDGs in the Times Higher Education Index. Today's topic is white nose syndrome versus Australian bats, featuring our two esteemed speakers, Dr. Nicholas Wu and Mr. Tomás Villada-Cadavid. Dr. Nicholas Wu, since joining the Institute in February 2022, he has been at the forefront of wildlife disease research. With a solid academic background from New Zealand and Australia, Dr. Wu brings a wealth of expertise. His current focus revolves around an ARC linkage project aimed at developing predictive models of vulnerability to white nose syndrome in Australian bat populations. Beyond academia, Dr. Wu actively engages in science communication, utilizing digital illustrations and graphic design to disseminate his research findings to both scientific and public audiences. Mr. Tomás Villada-Cadavid, a passionate advocate for bat conservation, joins us with a wealth of experience and expertise. His dedication to studying and protecting bat populations globally and contributions to bat diversity research have been invaluable. Currently pursuing his PhD at Western Sydney University, he focuses on generating spatially explicit models to assess bat vulnerability to white nose syndrome, employing innovative conservation strategies incorporating technology such as telemetry, camera trapping, and acoustic monitoring. Please join us as Dr. Nicholas Wu and Mr. Tomás Villada-Cadavid share insights from their cutting-edge research on white nose syndrome versus Australian bats. This thought-provoking discussion promises to shed light on the challenges facing Australian bat fauna and explore potential pathways for conservation and mitigation efforts. Please welcome.
Thank you for the introduction. Welcome everyone to our talk on white nose syndrome and bats. First of all, I would like to talk a little bit about bats. So first of all, bats are mammals, like we humans are. They're warm-blooded, they're hairy, their young feed on milk. They can see, hear, smell, and feel. You can see here on this picture on the bottom, Dr. Merlin Tuttle has used to train bats to go towards him and feed from his hand. And that way he could take pictures and investigate what vocalizations they were doing while going towards him. Here on the right side, we have a phylogenetic tree that is showing us how bats are related to other mammals. And you can see here how bats are more closely related to carnivores and other groups, including dolphins, cows, and horses, and not as closely related to humans and mice as people might think otherwise, because they're hairy and they look like mice sometimes, but they actually don't. And you can see here that bats are divided in two main groups, big bats and other old lineages called Jangochiroptera, which includes flying foxes. Oh, sorry, it's the Gintochiroptera. And then we have Jangochiroptera that was known before as microbats. Moving forward, we have the word Chiroptera. What does it mean? It comes from the ancient Greek. Kai means hand and pteron means wing. So it's a winged hand. And you can see here the morphology of the wing of a bat, how they use their fingers to support their wings. And you can see here the structures have been conserved or preserved in the different groups, also in birds. But they just elongated their fingers, or their fingers have elongated to allow them to fly. We have bats in very different sizes. We have very small bats that can weigh as little as 3 grams, like the bumblebee bat. And we have bats that can be as big as flying foxes with body masses up to 1.4 kilograms, which is massive. This is a good example of how we should not handle bats. If you find bats in the wild, please don't handle them. And as researchers, we should always handle bats using gloves as protection. Bat diversity. So worldwide, we have over 1,400 bat species. Of these 1,400 species, we have 81 bat species only in Australia. And out of those 81 bat species, 34 are found in New South Wales. You can see here in this map how bat diversity is mostly grouped in the neotropics. Especially in Colombia, where I come from, there is a high species richness compared to Australia and other parts of the world. We have over 215 species there. Bats come in all different shapes and colors. You can see here this bulldog-shaped face on this bat with this strange mohawk. We also have long snouts, and we have very long nose leaves and long ears. And all these characteristics and traits have evolved in different ways to allow them to echolocate sometimes, and also some characters as sexual, with sexual characteristics to attract mates. We have bats in different colors. You can see this diversity here. There is a wide diversity of feeding habits as well. We have fruit-eating bats here on the left side. We have nectarivorous bats that feed on nectar and pollen. We have insectivorous bats that can feed on insects that are standing on top of leaves. We have others that they can catch insects while they're flying. Others can feed on larger species like frogs and other vertebrates. We have species that can also fish. They have adapted, they have developed these big legs with big claws that they can use to hover over the water and catch bats. And we also have bats, very few, only 3 species worldwide that feed on blood. And they are only found in the neotropics. They can live in a wide variety of habitats as well. We find them in caves, which is the place where we are looking bats at the moment for our project, which Nikki is going to further explain later. They are also found in man-made structures like tunnels or bunkers. Sometimes they can be found in trees, roosting under the bark on tree hollows. They can also roost underneath leaves, which they modify by biting next to the middle vein of the leaf. And it folds and covers the bats, protecting them from the environment. Some other species have adapted to live and in this symbiotic relationship like this here with carnivorous plants. So they roost in there, and when they urinate or defecate, all this nitrogen can be used by the plant later while the plant is sheltering the bat. And we have other bats roosting from tree branches like flying foxes, which are very huge. This is the life cycle of bat. I have adapted this graph to show this life cycle of some bats for the southern hemisphere. So they go on this period of hibernation around May, and it ends around September. During this period of hibernation, in very cold climates, it's very important for them to gather enough fat reserves to overcome challenges of winter, such as food shortages. Then around September, they emerge from hibernation and they move to different places and maternity colonies to give birth to the youngs. and then the cycle goes on again. This is a compilation of books and resources that I would recommend to everyone that it's interested in learning more about bats. We have this nice book by Merlin D. Tuttle about pictures and curiosities about bats. It's a very general book. Then we have this very comprehensive textbook that talks about bat diversity and shows most of the species of bats of the world that we know up to this moment. And then if you're interested in Australian bats, we have this second edition of Australian bats by Sue Churchill. Hopefully there will be another edition coming soon. You can also find the first edition in the Western Sydney Library. And then there is also some good information about bats and other mammals in the Mammals of Australia. Of course, you can also visit the Ausbats website if you are looking for other information like distribution maps. It's also available in this website. So now I'm going to leave the stage for Nikki.
Thomas gave a really nice introduction about bats, the diversity that we have here, and the types of habitats that they occupy. Unfortunately, a lot of bat species are declining due to human influences. So habitat destructions, pollutants that they're releasing to the environment, and diseases that humans are carrying around as well that are passing on to bats. And one particular one that we have been interested in, especially for our Australian native bats, is white nose syndrome. Now, white nose syndrome is a lethal fungal disease caused by the pathogen Pseudogymnoascus destructans. We'll just call it PD for short, just to make it easier to explain. And PD was first introduced to North America from Europe in around 2006 or 2007. And it was detected in one of the caves in New York. And it was associated with population declines of bats there. And ever since then, it's been spreading all across the US and has been decimating bats all over. In fact, some populations have been associated with 100% decline, so complete extinctions of certain populations in the US. And it mainly affects hibernating cave-dwelling bats. And that's because the fungus itself is cold-loving and loves high humidity as well. So caves provide an ideal environment for the fungus to grow in. And because when bats are hibernating, they go into this physiological dormancy known as torpor, which means that their body temperature goes down and their immune system goes down during that period, which allows the fungus to grow on the bats. And that's what kills them, is that when they are infected with the fungus, they will often arouse from torpor because of that itchiness of the skin and also the inflammations as well that are occurring from the infection, which causes them to lose more body fat during winter, and they die from losing too much energy from that. How the fungus transfers between bats is through direct contact. So either by bats in contact with each other or by fomites, so objects or environments with the fungus on it can directly transmit to the bats. And the reason why it's called white nose syndrome is because bats that exhibit this condition often have little white patches on their nose and also on their wings as well. So it's very obvious to see when you see an infected bat. So because of this devastating effect that's happening in the US, people from all over the world have been trying to figure out how far has it spread. And there have been efforts in the Southern Hemisphere that have tried to detect the fungus. And so far, the efforts have been in Southern Australia and also across Chile as well. And what they have found is that PD, this fungus, has not been detected in the Southern Hemisphere yet. So that's a good sign. It's only restricted to the Northern Hemisphere. But there is a risk of it of Southern Hemisphere bats being exposed in the Southern Hemisphere because of the constant movements of people and, more relaxed biosecurity laws between countries in terms of washing your gear, shoes, caving suits, and so on. Australia is of particular interest because we have a very unique situation with our bats. A lot of them are endemic to Australia, meaning that they're only found in Australia. So they have a very evolutionary distinct heritage relative to other bats across the world. And we have a really good number of bat species as well here in Australia that are potentially at risk to white nose syndrome. Now, in 2016, there was a risk assessment workshop done by various conservation agencies across Australia. And what they concluded from this workshop is that it is very likely to almost certain that PD will enter Australia, and it is likely that the bats will be exposed to the fungus over the next 10 years. That's a very worrisome sign that PD is likely to arrive in Australia within the next 10 years. So there is a need to kind of understand how our bats will be able to respond and potentially adapt to this fungus. Will our bats be as sensitive as the bats in the Northern Hemisphere, North America, or will our bats be able to cope with this fungus if it does arrive? And so this is where this project comes into fruition. So it was funded by the Australian Research Council linkage grant awarded to Christopher Turnbull. And we, Thomas and I, have been brought on board to kind of answer the questions that are involved in this project. So the investigators include mainly the Western Sydney University team, but we also have got collaborative teams as well. Taronga Zoo, we've got the Arthur Wright Institute as well down in Melbourne. Lindy Lumstein is one of the most predominant bat researchers here in Australia, so her contributions have been very valuable. Got people from the University of New England, University of Melbourne as well, and these guys are mainly focusing on the immunological aspect of the bats themselves. We've also got intellectual help as well from University of Winnipeg in Canada, where at the moment a lot of the bats in Canada are dying off because of this fungus. And so Craig Willis is providing some valuable information on the most up-to-date research that's happening in the Northern Hemisphere as well. We've also got some partners as well that have been contributing to the project in some form or another, either in-kind contributions or intellectual or equipment support as well. And this includes the Australasian Bat Society, Taronga Zoo, Zoos Victoria, the Australian Speedological Federation as well, Wildlife Health Australia, New South Wales Government, Australian Government, and the Victoria State Government. So all of these partners have direct interest in this project and the outcomes from it as well. So what are the project aims anyway? Well, we've got 4 different aims that we're trying to achieve. One is looking at the potential exposure of this fungus to Australian bats. Second is the potential sensitivity of our bats if they're infected to the fungus. 3, developing vulnerability models to determine where in Australia they will be likely most vulnerable, and also provide some actions as well. So exposure, we're looking specifically at the pathogens themselves, so the traits, the habitat suitabilities, where they will most likely thrive in certain environments. Sensitivity, mainly looking at the hibernation biology of our Australian bats, because surprisingly, we don't have much information about just basic biology, especially during winter, of our Australian cave-dwelling bats. Very little information about that. So how can we tell how sensitive our bats are if we don't even have that start-off information? And this is where we're trying to fill in those gaps. From that, we can develop risk models, so areas where it's very suitable for the fungus to grow and where the bats have highest risk during winter as well, that we can develop from these aims, and also potential actions as well. For example, targeted biosecurity, instead of focusing all over Australia, maybe certain areas may be of research priority if the fungus does arrive in Australia. Provide information for cavers and also tourist groups as well that manage these caves about certain times of the year that cave access maybe has to be a bit more limited because of the bats hibernating in those areas and how much fat will they lose during that time. And also as well, because we're getting that basic information about the bat's health, we can also inform information for veterinary people or wildlife health carers as well on how bats normally go into torpor and how much energy they use up in normal environments and how they can also help bats recover for using that information. So what have we done? to address these aims? Well, we've got a whole bunch of different caves that we're examining across New South Wales, all the way from Mount Capita in the northern parts of New South Wales down to Kosciuszko National Parks as well. And for each of these caves that we're looking at, we've got a whole bunch of different things that we're doing. One is actually going into the caves and doing surveys of hibernating bats. So just going in there, counting them or taking photos of them to just get estimates of numbers and where in the caves they're hibernating. We're also sampling for PD in the environment as well. So we've got swabs that we go inside the caves and also swabbing the bats as well to detect if the fungus is actually already here or not. We've got samples ready to go, but we haven't analyzed them yet. So stay tuned for that. We're also looking at the cave microclimate as well. So addressing the first aim, cave exposure, potential exposure. So looking at cave humidity and temperatures as well. Also looking at activity of the bats during winter, and this can be done passively using acoustic detectors. So essentially putting a high frequency microphone outside of the cave and just measuring how often these bats are exiting and entering the caves during winter. And we've also got some time-lapse photography as well. So we've got silent camera traps that are placed inside the caves to measure how often these bats are waking up from their torpor as well. Some example caves that we have explored, just to give you an idea of the diversity and kind of the skill sets required for this. I'm not a caver by trade, and this is my first time doing cave work. So I get nervous and claustrophobic as well in some of the caves, and some of them require vertical descents down into that. But it's part of the job requirement, so I did have to learn it. But it was a fun experience learning these techniques. These are a series of photos that I took of Thomas going down Drum Cave in Bungonia. And you can see here he's getting some measurements of bats in this cave. Can any of you point out where the bats are in this section of the cave? Can you guys actually see where the bats are?
You can see droppings up there, the lining there.
Yeah, some droppings up. Ah, this pocket you mean? Yeah. What about this area? Do you see any bats here? There they are. These areas, these little dots here, are little clusters of bats that Thomas is surveying. So he's really good at this. Thomas has extensive experience with caving, so any difficult aspects, I kind of leave that to him. But yeah, some of our experiences here. Other ones, we actually have to go through tight squeezes and through streams as well. So we do get wet a few times going past some of the chambers. This is an example of me exiting one of the tight squeezes in order to get access to where the bats are. This is one taken at Mammoth Cave down up in Jenolan. Again, fun experience, not for the faint-hearted. And some areas we have to go quite a long way just to get to the cave itself. So one of the criteria is when we were picking the caves is that, first of all, it needs to have bats. But also, because we're deploying very expensive equipment in there, we don't want equipment getting stolen by people. So it can't be a very touristy cave in itself. And also, it has to be relatively undisturbed. So we've got a few caves up in the Kosciuszko National Park that requires a bit of a walk just to get there. Just to even access the caves. And some of them require river crossings as well. So there are some caves at Urungabilly that we had to get to. You can see the cave up here that we have to climb up on, and in order to access it, we have to go through a raft. Now we could actually just, use a rope and go down, but it's quite steep and it's on the opposite side. So we have to cross the river anyway, regardless. Okay, so some preliminary results that we have. This work is still ongoing, and we're going again to our sites this winter to continue our surveys in some new sites and visiting some regular ones as well. But here's some preliminary results that we have from last year. So what are the potential exposure risk of PD based on the cave microclimates that we have? So this is just a reminder, PD grows in very cold environments and very high humidity. So temperature-wise, they ideally like to grow at 13.5 degrees with plus or minus between 11 and 16 degrees. But they in fact can grow between 0 to 20 degrees. So that's their potential range of growth. And also high humidity. So anything above 80, about over 70%, they will grow in. So how do our caves look compared to this? Here's a few example caves with the temperatures on the x-axis, so the range of temperatures that we measured inside the cave. And we went in every 10 meters inside the cave and just took temperature and humidity measurements and just mentioned whether or not there were bats present when we were taking those measurements. So the highlighted colored one, so yellow here, shows where bats are present when we're taking the measurements, and gray is when the bats are not present. It's the same with humidity as well. So we've got caves that have very high humidity, for example, Cormorant Cave and Carcass Cave up in the Kosciuszko National Parks, very high humidity throughout, while certain areas, we also have artificial ones as well. This is Coal Loader in North Sydney that I've known to have bats in there. That's an open-ended tunnel, so it's not one-ended, which means there's a lot of airflow going through. And so the humidity in that particular tunnel is relatively lower than natural caves itself. But if you have a look here, where the optimal temperature that PD can grow in, you can see that it overlaps with a lot of areas that bats like to roost in. This is a worrisome sign. And it's the same with humidity as well. Basically, almost all our caves have suitable microclimates for the fungus to grow in. So if it does arrive in Australia, at least for these caves, there's a high risk of exposure for the bats if the fungus does arrive. Here's an example camera trap recording of the bats, for example, in Mammoth Cave. So what we've did essentially, here's the cave entrance. We walked through, got a few measurements of temperature and humidity on the way to where the bats are. This highlighted section is where the bats are roosting, and we put a camera trap there just to leave them and see how often they arouse from torpor. So here's an example video of them arousing. You can see the time here. It's measured every hour, so there were photographs taken every hour, and the days and the temperature as well inside the cave. And you can see that the clusters, there are a few individuals that move around a lot, but overall there are clusters that stay there the entire time and barely move at all. So they tend to stay in torpor for quite a while inside the cave. That's just to give you an example. And some of the issues that we have with these camera traps is that droppings occur. If you notice a bat just dropped poo on the camera lens, and so we can't get really nice recordings after that. So one of the issues with camera traps is that the lens can get dirty very easily inside caves. So getting accurate measurements of individual bats arousing has been proven difficult, but we're finding ways to try to mitigate that, putting shields on top or something, or finding ways to kind of make it a bit clearer. Troubleshooting with field work. Okay, what about potential sensitivity? Now with that, we need a little bit more detailed information for specific bats because of the amount of work required for this. So there are a few sites where we measured the body conditions of the bats, which is their body weight and their forearm length as well, for a few different areas. And that represents how much energy they use during winter. So we measure body condition across the winter period, and also measured individual torpor and arousal for bats. And Thomas will be presenting that section of the results. So I'll be mainly focusing on the body condition part. But we only focused on two sites, one in a warmer site and one that's considered a cool site, because we predict that the cool sites would be, bats in the cool sites will be most at risk because they will either stay in torpor for longer, and that they will use up more energy during winter because of the cold conditions that are being experienced. So how do our results look? We measured body mass at different time points. We measured that at late summer around February, start of winter, middle of winter, and then at the end of winter as well. Now with these data were separated by males and females as well, with the females in the blue and the males in the black triangles. So you can see here with the cold site, the bats in Jenolan, the cold site, tend to lose more body mass during winter than the warm site. And there is a sex difference as well. Females on average tend to lose more body fat than males across both sites as well. You can see here females in the cold side lose 30.8%. Females in the warm side lose 14.7%. Okay, so that gives us some information about how much energy they use up during winter. How does that compare to other bat species in North America? So we've got a comparison with other hibernating bats in North America as well. So this is the percentage of winter mass loss relative to the mean surface temperature of the environment. So the warmer the environment is, the less body mass they will lose on average. There's still quite a bit of variation. But the important takeaway message here is that our bats, so Janolan, yes, about here, and we also got some data from Portland as well. So this is the southern bent-winged bats from one of our researchers from University of Melbourne who collected that data. We've also included that as well. And our bats lose the same amount of body fat as the North American bats under the same kind of climate conditions as well. So if the bats that are vulnerable up in the northern hemisphere, the US as well, if we're assuming that fat loss is an important factor for their sensitivity, our bats would be equally as sensitive based on similar fat losses during winter. So now Thomas will talk a little bit about his aspect of the study, the temperature sensitive telemetry, and he'll explain more about it now.
Thanks, Nikki. So we have seen that there are in differences in body mass between warm and cold places, as Nikki has shown before. At cold places, they gain a little bit more body mass, and at warm places, they don't gain as much body mass pre-winter because they don't have to face such difficult winter conditions because the winters are milder in warmer areas in our study area. I was interested in looking at torpor patterns. So let's talk a little bit about torpor. It's that period of time where bats go into hibernation. They drop their body temperature, save energy, which also drops their metabolism. And that helps them go through the winter. In order to investigate that patterns of torpor, we are measuring skin temperature of the bats throughout the winter. And to do that, we use these little transmitters, VHF radio transmitters that pulse at different intervals depending on skin temperature of the bats. And we collect all that data on these loggers that we have deployed inside the caves with little antennas that will receive all the pulses intervals that we will then transport into temperature readings. This is a picture of Nikki and Chris capturing bats at Janolan caves. We use these hard traps, which bats cannot detect fast enough, and they crash against them. But it's nylon, and it's very soft, and they don't harm themselves. And then they fall into these bags, and then we pick them, and we attach the transmitters, and we release them on site. So we can record them at the chambers where they are roosting during the winter. This is a video of a bat being released. 3, 2, 1. And here we have a picture of me trying to detect the bats with this antenna inside of the cave to make sure that they are roosting in that area, and then deploy the stationary antennas that will help me record those pulse intervals. This is some of the data that we have gathered so far, and this is like a sneak peek of what we will be presenting at the ABS conference next week. Nick is going to be presenting some of the results that he already shown, and I'm going to be presenting in particular individual torpor patterns of bats in New South Wales. So you can see here there are differences in cave temperatures already. We have Mammoth Cave and Paradox Cave, which are the caves in the cold places. Here we have external air temperature on blue and green and yellow on the top. There is quite variable, and you can see that temperature drops to the middle of the winter, and we can see cave temperature here, the red line, and it's very stable, and it's not very different in the cold areas compared to the warm place where cave temperature is a bit higher. There is also a bit more fluctuation, and the mean daily temperature on average is higher compared to the other sites. This is some of the data that we have collected so far. We have some gaps on our data because we have some difficulties with the transmitters and the receivers, but overall it's a very good pool of data, and we will be joining this data with data that has been collected by master's students in the previous years at Genoan during that particular period of time, so it's very suitable for us. You can see there are different torpor patterns. Here on white you can see skin temperature of the bats, and when temperature drops it means that they're going into a torpor bout. And you can see that torpor bouts here can last for several days. On shade, in the shading area we have nights and then we have daylight here. They go for 1, 2, 3, 4 days of torpor. They can reach up to 6 throughout how we have measured. And sometimes they have been measured to reach up to 13 days in torpor at Genoan. You can see here on blue, that's the external air temperature. So it's quite cold, but the cave temperature, it's stable and it's always at 10 degrees Celsius at this cave. You can see here there are shorter torpor bouts recorded as well. It's not only long torpor bouts during this period of time. So it changes, and it changes also depending on the individual, whether they're roosting probably in big groups or not. And you can see here at Yesava, the warm side, they're very, very active. So they don't go into these torpor bouts that often, and not for as long as they would at Genoan. So we are already seeing some differences. We can see here on this plot, on the blue dots, that's the cold side at Genoan, and the warm dots, the yellow dots, it's Yesava. And this is on the y-axis, we have torpor bout length. And then here we have the time at which they enter in torpor. So during the middle months of the year, when it's colder, they were using torpor for longer periods of time. And you can see that there is a big difference in between the warm side and the cold side. At the warm side, they were not using torpor for more than 2 days, and mostly for 1 day and less than 1 day. Whereas in the cold side, they were using torpor for up to 6 days. And when we bring the data that has been collected before, we can see the trend amplified. And it is telling us that they are even having torpor bouts up to 13 days. And we have this pyramid here, like this shape. So there's shorter torpor bouts at the beginning, and then it increases, longer torpor bouts when it gets colder during the middle of the winter, and then it goes down again, and they have shorter torpor bouts when it's getting warmer and they are ready to go and feed out again. It doesn't mean that they don't feed during the winter, and we have seen that. They are also active sometimes during the winter, and they can go and feed because there are resources. There is also a pattern here in the time, regarding the time of arousals. We can see that they often arouse overall, both sides. They overall arouse just after sunset. And when they arouse, it's like 15 minutes after sunset. And when we see and we look at difference in between the two sides, we can see that there are not big differences at the time of arousals. And it seems like circadian rhythm and what they have been like adapting for years and ages, it's to these rhythms of daylight and nighttime. And they all usually arouse at the same time. And that doesn't change depending on the sun. So far, that's been what we have been working. There is more to do. We're working on analyzing this data. And we're looking forward to your questions. If you want to know more about this project, you can go to Bats Lab website. And thank you.
So it looks like we've got a lot of questions online. Maybe we'll start with people in front if they have any questions now, and then we'll move to online ones. Yes. So one of the questions, this is for the online folks. One of the questions is, is there a age difference in susceptibility of white nose syndrome that's been seen in the Northern Americas, right? So far, it doesn't seem to be age specific. It is size dependent though. So smaller bats tend to be more susceptible to white nose syndrome because they have smaller size, they have less fat to start off with. So across winter, they will use up more proportionally than a larger species. And so by that deduction, if a juvenile is smaller than an adult, they would be more susceptible to the white nose syndrome than an adult as well. But what studies in the US show that doesn't seem to be much of a difference. It seems like both juveniles and adults are equally likely to be susceptible to the fungus. So are there certain attributes for different species that make them more susceptible to the fungus? And yes, there is. So species that are obligatory cave dwellers are more at risk of white nose syndrome. So for those who are more general, they will roost in trees, man-made structures, and caves. Only the cave populations or ones that roost in caves are more likely susceptible. So depending on the roost type, that's a big factor when you're looking at white nose syndrome susceptibility. Also, differences in how often they arouse as well. It seems like the ones that stay in torpor longer are more susceptible because it allows time for the fungus to grow on the skin for longer. If you think about it, when they arouse from torpor, their body temperature goes up as well, right? And it goes well above what the fungus can grow on. So when they're aroused, the fungus doesn't grow on the bats because it's too hot for it. But when they go back to torpor, they start growing again. So when bats are in torpor longer, they're more at risk of gaining more of the fungus itself. There's a whole bunch of different factors, but just giving you a few examples. Basically, the more energy they use up during winter, the more risk they are. And different species have different winter energy uses for that. So the follow-up question of that is another attribute, which is clustering. So bats that cluster together are able to spread the fungus more. Doesn't usually associate whether or not they're sensitive to it. It's just whether or not how many individuals will be infected. So we do have some species here in Australia that roost individually, Rhinolophus, for example, or in very small clusters. The reason why we focus on Miniropterus is because they have very large clusters, up to thousands, 10,000s in one cluster at once. And so they're most likely to be susceptible because of how fast the fungus could spread. If it reaches one cluster, it will spread across all those individuals. Yes?
In the Northern Hemisphere, can we say that it was people doing caving that were the main spread of the disease? And if that is the case, does that mean that we have to put all our cases in Australia to make sure that their equipment is sterilised?
It's a controversial topic. So one of the questions was the fungus that was introduced into North America really a caver? Was it from a caver? And if so, do we need to talk to cavers here about preventing, being a bit more health conscious about contaminating soils, for example, across different sites? We don't know if it exactly was a caver because the cave that it was detected in was considered a tourist cave. We can't say that it is a caver. It was just someone from overseas in Europe brought it over, had some contaminated shoes that entered in and then contaminated the cave. It is likely that it's a person that goes to caves often because they would have had to go into a cave in Europe in order to get it onto their shoes, go into North America, and then go to another cave. So that's the best we could say. So we can't say it is a caver. It's just someone who goes into a cave a lot. Regarding Australia, we do talk to a lot of cavers here, especially with the Australian Speleological Federation as well, about white nose syndrome. So they are aware of it. And they know about the procedures of just being careful when you're bringing equipment between different sites. Cleaning equipment is very time consuming and sometimes dangerous as well because the procedures used to actually sterilize equipment can actually damage it as well at the same time, makes it less safe for cavers to access. So for example, ropes that have been soaked in very hot water or chemicals in there that can soften it up is actually not good for the ropes and the safe. So there's a balance there that we need to consider as well. But the folks in North America seem to have got it in terms of the safety precautions required because the fungus is already there. So they're finding ways to reduce the spread to other caves that haven't been infected yet. And so we're referring to Australian caves here to kind of follow the same procedures that they're doing in North America. Should we take some online questions?
So there are some.
There's one in the room? Oh, yes. Yeah.
What we should do find that we think has white nose.
Yes, absolutely. So in the So if someone does find a bat that is sick, that is potentially infected with white nose syndrome, that you think might be infected with PD, the best advice is to contact Wildlife Health Australia. There is on the website here that's provided, there's a specific section on white nose syndrome and information about it. You contact them about a sick bat. In fact, if there's any sick bats, regardless if you think it's got white nose syndrome or not, let them examine it. And they will do is swab it and examine it, the other features as well, and determine whether or not the sick bat is due to the fungus itself or other health factors. So in general, just contact Wildlife Health Australia about this. You want that, or do you want me to?
That's a tough one.
It's a tough one.
Yeah, it's a tricky one. I can answer this one. So that's a very good question. It's not clear to me how this El Nino oscillation would affect white nose syndrome, because the main driver of the disease would be first that bats are exposed to it. So if it comes to Australia, I don't think El Nino would have any effect. It's mainly about biosafety management. And then if it gets into the caves have such stable environments that if the microclimate is suitable inside of the caves, the external climate won't have such big influence. So in that sense, I believe that El Nino is not having a big impact, or is not going to be a big driver, whether the disease is introduced and the bats are vulnerable to it or get infected by the PD. Okay, so Wildlife Health has a plan to respond to this emergency, and how this is going to be done. It's stated in the management plan.
Yeah, the management plan is quite extensive. We don't know how detailed we want to go into that, but there is one, and we've been part of that discussion group as well on how to respond if PD does arrive in Australia. So there is a plan. Yes.
There's a comment in the chat that there's a WHO website is there, and there are resources that explain what to do if you find a suspect case.
Perfect. Yeah.
Someone shared them. Someone shared Wildlife Health Australia website.
Yes.
Yeah. So thank you.
Thank you for that.
And Erica got one more question. If a bat arouses, does the increase in body temperature kill the fungus or just send it dormant?
Yep, can answer that one. So it won't kill the fungus necessarily. It will probably reduce the reproductive cycle of the fungus, so it won't grow as much when the bat is warm. But during that time, the bat will have time to arouse, and the immune system will kick in and will start trying to remove the fungal growth from face and wings. So it's important for bats to do this when they're aroused, but at the same time, is if they arouse because they are feeling itchy, and that's going to consume energy reserves. And that's what's very, very negative for the bats, because their arousals take up a lot of energy. And when they use this energy and there is nothing to eat outside, they could probably not make it to the end of the winter. So in other words, yes, the fungus will not die because the skin temperature rises, but it can help bats get rid of the fungus if they arouse. But that could be negative for the bats as well. So there is like a trade-off.
There is another question. Is everyone doing similar research in Africa?
As far as we know, there is no one doing research on white nose syndrome in Africa. There is research being done in South America. They have tested whether there is PD, and they have found that so far the places they have screened, there is no PD. And it has been done in Australia before as well. There was a study, they were screening for PD in Victoria, and they didn't find it. And now we're trying to collect more samples for New South Wales and make sure that we don't have it yet. Okay, that's a variable that I haven't been able to measure yet. But the periods of activity are very, very short. They can be of a couple hours, not more than a couple hours. But sometimes it's very difficult to measure that, because a bat can be active, can arouse and fly away and not come back to the same site, but go to another spot where I can't detect it with my antennas. And it doesn't mean that the bat, because I'm not recording it, and it comes back again like 2 days later, but it has been active for 2 days. It just means that I was not recording that bat and what it was doing during that time. But the periods of time where they have been active, that I can, with a high interval of confidence that they have been active and then they did something else like going to torpor, has been very short. And that's still to be quantified, and I'll hopefully be able to provide numbers in the future, but it's very short, just a couple of hours. There is definitely insects available, because they do arouse and they go out, and we don't find big changes in population sizes, right? And they do arouse quite a bit. But it's probably different in between areas. That's something we haven't quantified in this project. But the proxies that we use to tell if there is insect availability is how much they arouse. And it is very likely that they arouse because there is insects. And we see them flying outside of the caves, even during cold days in winter, which is probably them going out to feed. So that is indicating that there is food available. Probably not as much, but there is.
So the question is whether or not food availability, seasonal food availability, should be considered as a factor when looking at arousals of bats infected with white nose syndrome. So evidence from North America is that there definitely is a stronger seasonal variation of food resource because winters are much more extreme than North America, so there's barely any insects around. So they go into hibernation a lot longer than our Australian bats. And they arouse more often when they are infected because they want of that uncomfortableness of being infected. So they're arousing to get rid of. So in fact, I'll give you an example. Some caves in Canada, the exits or the entrances where they usually fly off is completely covered by snow or ice. So they can't even leave at all. But there's been evidence that they still arouse from torpor, and that's mainly because they're trying to combat the fungus itself. But because there's no food around to feed on, they can't replenish that energy back compared to bats that are not infected. So if it happens here in Australia, if there's still enough food availability around and they are infected, they can arouse, go out and forage. And if they can get enough food while increasing that immunity up to fight back the fungus, they should be able to do okay. It's mainly about that energy balance, whether or not they can afford to lose more energy from fighting back the fungus. So if there's a particular bad year, like drought or some period of extreme coldness, that will have an effect, yes. Yeah. So European bats seem to do well, coexist well with the fungus, right? We don't see any population declines associated with white nose syndrome in there, even though they can be infected. And people have looked at that comparison between North American bats and European bats. And their immune system, when they are infected, seems to be quite different when they're infected. So there's some evidence of coevolution, coexisting with the fungus. So probably when the fungus arrived thousands, millions of years ago in Europe or evolved, the bats would have populations would have slowly adapted to that, while North American bats are completely naive to it. So their immune response kind of goes into hyper overdrive to the fungus. So even though the hibernation biology might be similar to each other, the immune system reacts differently to the fungus itself. And so one of the research that we're doing here as well in collaboration with University of Melbourne is to see how bats' immune responses change in response to a generalized infection, because of course we are not going to risk bringing the fungus here and infect them in the lab just to test that out. So we've got other ways to approximate sensitivity based on immune responses to other types of pathogens.